to go slowly, and hopefully people will not be overwhelmed. The state of threat, and then follow up with some suggestions for how we interpret some of the definitions, some of the concepts that are in IUCN, and interpreting them so we can do that with fungi. And then lastly, we'll break into these groups and actually practice doing the evaluations. So what is fungal conservation? Basically, it's an interdisciplinary activity that combines science, communications, public relations, land planning and planning, and the political, engaging politicians for engaging policy. And its goal is really to raise awareness of the importance of fungi, the fact that fungi need to be included in conservation discussions, in conservation actions, and have access to funds, to monies that are being put forth for conservation activities for animals and plants. It needs to be animals, fungi, and plants. I don't have to tell this group, but fungi are important. And really, life on the planet as we know it wouldn't exist without fungi. But fungi are, is this bar bothering people? Is this in the way? I can't tell. Anyway, but so fungi are not immune to the same threats that face plants and animals. They are threatened by loss of habitat, by pollution, by overharvesting and other forms of overexploitation, by climate change. So a step to be actually to effectively conserve fungi, one of the first steps is determine what species are doing well, what species don't need assistance, versus what species are rare or in a risk of going extinct, because we never have enough money, we never have enough time, we never have enough people. So we need to prioritize our activities to make sure that we're focusing our work on those species that are most in need. And that's usually done through a process of red listing. Now, unfortunately, the most fungi have not been evaluated. We don't know the risk of extinction or the conservation state of most fungi. And this has caused a problem. Not being on national and global red list has made it difficult to include fungi in discussions and actions of conservation, and also made it difficult and may oftentimes to obtain funding to do our work. We're not included in programs that provide funding for conservation work. But we are making progress. So in 2013, there were only 3 fungi on the IUCN Global Red List, 2 lichenized fungi and 1 mushroom. Now there are 408 species, and as Ida mentioned, 139 of those are species that occur in Latin America. And workshops ongoing and future workshops are going to significantly increase the number of species included. So here's just one plate showing some of the Southern South American species that we have, whether it's this beautiful bolete, or I know that Ricardo would think this beautiful resupinate fungus, this Rhytoporia, Cetaria, so we have ascomycetes, and lichenized ascomycetes is this lichen from the Galapagos. So I guess one of the first questions people often ask is, what is a red list? What is a red list evaluation? And so basically, it's an evaluation of a species going extinct utilizing a comparable and objective method. So the evaluation estimates the potential change in population size of a species over time, with the idea of inferring its risk of going extinction, right? So if a species population is increasing, or if it's stable, it is probably not in a risk of going extinct. However, if that population is declining rapidly, it is probably not going in the right direction, and it could be at risk. So the IUCN has developed a series of criteria to basically rank the threat category of a species. So currently, most fungi fall into this. They have not been evaluated yet. Then there are a number of species that we have tried to evaluate, but the data just are insufficient. They're data deficient. And these are usually species that maybe is only known from the type locality. So we can't really say anything more about it, but that it was recorded one time from one location. And there are a few cases where the taxonomic confusion is so high that we don't really know how to evaluate it. But for species that we can evaluate, a large number are going to be least concerned. They probably are doing okay. We don't have to spend a lot of effort trying to conserve these species. They're doing well. But then there are a group of threatened species, and these can range depending on how intense the threat is. They can be vulnerable, endangered, or critically endangered. We do not have any listed fungi that are extinct. There probably have been extinct species, but we don't have any listed. Or extinct in the wild. Extinct in the wild would be species that only occur in culture collections or something like that. They no longer occur in the wild. And again, we don't have any of these species as well. But as you'll see, we have a number of species that are either vulnerable, endangered, or critically endangered. Now, it's important to remember that just being on a red list is not sufficient by itself to establish conservation priorities. Developing conservation priorities really takes into a large number of features. A very important one should be the risk of extinction. But there's other biological factors, social factors, economic factors. And all of these are considered in trying to decide and weigh the importance of these different criteria, and then they go into setting the conservation priorities for a region, for a country, for whatever. And just as an example of that, on this side, this is the Tropial. I don't know if we have anybody from Venezuela, so you can correct me if I'm wrong, but my understanding is the Tropial is the national bird of Venezuela. It is least concern. It really doesn't have a high conservation threat. But there is a lot of effort and funding going to maintain the population of this bird because it has so much social importance. And you can contrast that with the Anopheles mosquito, which carries malaria. And I don't know of any study that's going to protect this mosquito. In fact, the funding is going to eradicate this mosquito. So these are examples of where the social values, health values, whatever else, take precedence over a risk of extinction. So what are some important reasons for creating fungal red lists? First off is, so we all know that politicians, land managers, people like that, they're paid to make decisions whether they have the correct data or not. They're going to make a decision. So it is important that we make sure they have the best data available to them, so hopefully they will make the correct decision. There's no guarantee that if we give them the right data that they're going to make the correct decision, but the likelihood that they do, that they make a decision that is correct when they do not have the data is very low. So it's important to get that information out. Also, red lists really communicate the value and the recognition that fungi are important, that they are in need of conservation, and this is to politicians, conservation organizations, other interested parties, including the general public. So every time there is an update to the IUCN Red List, there is a lot of press releases, there are stories in newspapers, and so it's a great opportunity to highlight and to publicize the importance of fungi. And as one is doing these assessments, we identify gaps in our knowledge. It might be taxonomic problems or lack of knowledge about its distribution, or identifying what the threats are. And so this process of red listing goes at the same time as understanding the biology and the research needs of the species. So they work together. And lastly, but very important, not having fungi on red lists gives the incorrect idea that fungi aren't in need of conservation. And they don't need funding. They don't need to be included in conservation action plans because, again, politicians, land managers, conservation biologists use the list and what's on the list oftentimes to make these prioritizations. So if red listing is so important, why aren't there more fungi on it? And that's because there are challenges, right? As in many things, important things are not necessarily easy to do. So there is some basic data that one needs to be able to carry out a red list. We need to know what the distribution, the biogeography is of the species, right? If you don't know where a species occurs, it's really hard to talk about whether it's in need of conservation or not. We need to know something about the population size, right? Remember, we said that a red list is the evaluation of the change in population over time. So we need to be able to estimate what is the population size of the species, and then also infer or estimate how that population may be changing over time. And then over what time scale is this change going to take place? Is this a very short-lived species, or is it a very long-lived species? And then what are the threats? It's nice to say, oh, this species seems to be going extinct. Why? What are the threats? And when possible, it's great if we can make some recommended solutions to mitigate against those threats. So we need those kind of data. And as we all know, with fungi, it's sometimes difficult to get them, right? First off, there are a lot of species of fungi. The Hawksworth and Luking paper is probably now the most accepted estimate of the number of species, somewhere between 2 and 4 million species of fungi globally. But it's not just species diversity. Remember, we have so much taxonomic, phylogenetic diversity, morphological diversity, ranging from single-celled like chytrids to multicellular organisms like our ascomycetes and basidiomycetes. And so it's not just morphological variation, but ecological variation, from aquatic fungi to fungi that live on land or whatever, from saprobic to pathogenic to symbiotic fungi. So a lot of diversity we're dealing with. And it's not just at broad layers. I work with Agaricomycetes, and so even within that group, right, we have traditional mushrooms, we have coral fungi, we have bracket fungi, we have stinkhorns, earth stars, and these things vary from ectomycorrhizal species to saprobic species to pathogenic species. Excuse me. So a lot of taxonomic morphological diversity that we have to deal with. And at any one spot, there are a lot of species. So this is an older picture. You can tell because of my hair color has changed over time. But this picture was taken in China, but it could have been taken in Brazil or Colombia or Peru, where you go out, and this was from a 10th hectare plot where we spent the day, and we collected over 100 species of mushrooms in that 1/10 hectare plot that day. And the next day we went out to not that far away, to a different mountainside, and we collected again 100 species, many of those different from this 100. So we have very high alpha and very high beta diversity as well. And, we're finding these new species by field work. This is Macrocebe tetans from Costa Rica. But most of our new species now are being done as we do modern revisions of species using molecular data. So, for example, in North America, until 25 years ago, there were 2 species of orange chanterelles, one of which was Cantharellus cibarius. We now know that Cantharellus cibarius doesn't even occur in North America. It's a European species only, and there are 35 species of orange chanterelles, mainly identified now by DNA data. But probably the biggest challenge is not just the numbers, not just the diversity of fungi, but it has to do with the biology of fungi, right? Most of the organism long-lived part is the mycelium growing in substrata, in this case in leaf litter. And when conditions are correct, it'll put forth sporocarps. And in this case, because we know that these are all grown from one individual, all these mushrooms are coming from one individual. But when we're in the field, it's more difficult. Is this one individual? Is this 2 individuals? Is it 100 individuals? How do we count these numbers. That has been a big challenge. But there are also now opportunities. We've had a lot of progress in generating new data that allows us to begin to do these red listing. So in the last 50, 60 years, there's been a lot of field work that have documented new species and distributions. We've got a whole new generation of young, enthusiastic mycologists throughout, say, South America, throughout Brazil, many of them sitting on this Zoom call. So it's great. So we're finding a lot of information from the field. And at the same time, there's new and growing data based on laboratory studies for understanding population genetics, understanding the biology of these fungi. So we're starting to generate the data that we need. At the same time, the mycological and conservation community are realizing the importance of including fungi in conservation action and are interested in conservation of fungi. And then recent bioinformatic tools, databases, have really enabled us to access information in our fungaria, and also have enabled and encouraged amateur mycologists, amateur naturalists to provide information on the distribution of fungi. And it's really making a lot more information available for us to use. So just some examples. Ida mentioned, we now have international societies. So this is the International Society for Fungal Conservation. I know that the Latin American Mycological Association has a large fungal conservation committee that is actively working to actually develop a strategic plan for fungi in South America, Central America, and the Caribbean. So very exciting work. And in other parts, in Australasia or in China or in Europe, there are these groups that are focused now on fungal conservation. Ida mentioned that the International Union for the Conservation of Nature, IUCN, has now recognized the importance of fungi with multiple specialist groups, and the recently formed Fungal Conservation Committee that helps coordinate all of this action for IUCN and for groups that partner with IUCN. And then really importantly, we're starting to see other types of organizations being formed. So non-governmental organizations. I think the first was Fundación Fungi, Giuliano Furci's, but we now have, Fundación Fungi Cosmos in Argentina. We have other NGOs being formed around the world now. And so having these dedicated organizations to assist with this work, to assist with finding funding, to assist with popularizing, communicating about fungi is very much, educating people about fungi is very, very important. So I mentioned we now have new tools to look for data that are in our fungarium. So this is the North American Mycological Collection Portal. So we can go to this site and search all of the fungal collections and fungaria in the US and in other participating countries. Something like iNaturalist. So here's how amateur mycologists, field biologists, just people interested in nature are contributing. So I checked last night, and currently there are 3,550,000 observations of fungi on iNaturalist, representing basically 15,000 species. And what totally shows the power of this, there are 323,000 people that are posting pictures of fungi on iNaturalist. 323,000 people. How many mycologists are in Brazil? Trained, professionally employed mycologists, or in Colombia or whatever. If we can figure out how better to engage these 323,000 people, we'll have a much more data to be able to do our work. And so iNaturalist is just general, people go out, see an interesting fungus, they take a picture, but there are a growing number of projects to focus these efforts. So two of them, one of them is in the UK and in Britain, so the Lost and Found project, where these naturalists were asked to go out and search for fungi that hadn't been seen in England or in the UK for a number of years. The thought was they were either very, very rare, or they might have even been locally extinct, extirpated from the islands. And what they found was some species they found were much more common than they thought. But there were other species that had not been reported and still are not reported. So they have good data to say that these things probably are lost from the island. Fungi Map is probably one of the first of these programs. And what Fungi Map is in Australia is they came up with a list of 100 species and asked these field naturalists to go out and document where they find them. And so they were actually able to get then detailed distribution maps of these species. And they've now moved on to another set of fungi to continue this work. So really exciting. And so that's based on specimen data. And we know that there is growing information. I need to update this slide because it's 2011, but using environmental sampling, using DNA data to map the distribution of fungi. And this is doing a great job of adding to our understanding of the distribution of fungi. So taking all of these data together and these opportunities, my colleague in Sweden, Anders Dahlberg, and I, now 10 years ago, wrote this paper where we made suggestions on how to interpret the criteria, the definitions that are in IUCN, and use those for red listing fungi. And we'll be talking about these suggestions for most of the day today. And at the same time, we started this global fungal red list, which is a website where people can nominate species and add data on those species, which will then be assessed. So here's the front page of the red list. Then each species has a species page. This is the page for Citaria exigua. Part of the page where people can put information on the geographic range, population trend, habitat and ecology, photograph, etc. And then we'll take those data and we'll have a workshop to discuss that, refine it, and finish the assessment. In the old days, we used to get together in person. And so this was actually our first Red List workshop. It was done in Sweden, but you'll recognize some colleagues with Joaquin Cifuentes and Mario Aschenberg and Esperanza Franco and Juliana Furci. I think that's most of our Latin Americans on this slide. But of course, more recently, we've been doing these virtually. And so this was the workshop that Ida mentioned we did last year, focused on Southern South America and Colombian material, but we had some Mexican material as well. Very effective workshop. And we'll be planning another Latin American workshop coming up later this year. So today is more of a training session. Those of you that want to participate in these future workshops, let Ricardo know, because we'll be pulling one of these together again in the future. So in conclusion of this part, then we'll talk about details in a bit. We know that some fungi are threatened and are in need of conservation. And we now have a growing set of data that allows us to evaluate the conservation status, the risk of extinction of fungi, using the standard process that is used for plants and animals. In other words, the IUCN Red List. But also remember that red listing isn't the end of the process. What we do is we take the data that we have from the red list, and importantly, the enthusiasm and community that we've developed in that process to identify conservation action and implement those actions so that we can actually reverse the loss of these fungi. So we can actually use the data that's in the red list to conserve these fungi. So that's the first part of my presentation. I don't know if we want to take questions for this or take a small break. What do you want to do, Ricardo, for this before I go on to the next section?
Greg, up to now, we do not have questions. So you can start your second part of your talk.
Okay, I will do that. Let me go to share my screen. Go back to here. Oh, we're going to end that slideshow. Okay, so what we're going to do now, we really talking about why we would red list and some of the data and why we can now red list. So this section is going to be more on the details on how we actually do the work. So I want to do two parts in this. One is go over the categories and the thresholds that we use to evaluate the state of threat. And then spend some time going over the suggested ways that we interpret IUCN definitions within a fungal context. So, what species can be going extinct, or what species do we need to worry about? We usually think of only rare species, but it's really also they can be very common species that are in decline. They can be infrequent species that are in decline, or species that are very rare. So it's not just the rarest of the rare species, but it can be other species that we need to worry about that may be at risk of extinction. And my example of this are, these first two are from North America, but I think they work. So the passenger pigeon was my understanding was the most common vertebrate animal on the planet. There are stories talking about when the flocks of this bird flew overhead, it would darken the sky for hours. There were millions and millions and millions of individuals. And in a fairly short time, they were hunted, they were shot to extinction. And the last bird died in a zoo in Ohio in the 1920s, I think. Species is extinct, but yet it was super, super common. The American bison had kind of a similar story, right? There were millions and millions of individuals, and they were hunted almost to extinction. There were only a couple of hundred animals left. But there were some conservationists that recognized we do not want to lose this animal. And they figured out what the threat was. It was hunting. Figured out what was a conservation action, stop hunting, and develop a breeding program. And now the species is thriving. In fact, they are being hunted again because of the success of this conservation program. So these were both examples of very, very common species that, in this case, went extinct. This one almost went extinct. And you can contrast that with something like this hidellum, which is pretty rare, but in decline, or this orchid, this ghost orchid, which is very, very rare and known only from a very small part of the world. So all of these, even though their population size, their biology is very different, all have population or conservation importance, and all had threat of extinction. So IUCN uses 5 criteria to assess the threat of extinction. So Criterion A is basically looking at the rate of decline of a population. So there is a certain number of individuals, and over time, it is drastically being reduced. Okay, so this would have been a great example of the passenger pigeon or the bison. Okay, there were a lot, but they declined quickly. And see if this line continues, they go extinct. Criterion B is something like that orchid I was talking about that has a very restricted geography, a very restricted distribution, and it's in decline, or it may be a fragmented population. So this is just doesn't make a difference how broadly the species is distributed. It's all about the decline. This is for species that are very narrowly distributed. Criterion C is similar to A, except for the fact that you've started with a small population, right? So your line of decline, your slope of this line is much less than here, because here you start with a lot, so you have to decline quickly to go extinct. Here, there's not a whole lot of individuals, so even a slight decline may cause it to go extinct. Criterion D is just that you have a really, really, really small population. And so this is a case that it may not be in decline, but the population is so small just from random events, from stochastic events, a hurricane, a fire, a construction site could wipe out that whole population. So it is threatened just because it's such a small size. Oops. Don't want to do that. Don't look. And then the last E is quantitative analysis. And virtually no organism, plant, animal, or definitely no fungi have been analyzed. You got to know great details on its population, size, and be able to calculate how that's changed. So it's one of the criteria, but nobody uses it. And then within each of these criteria, there are thresholds. So remember, Criterion C is the criterion that has to do with the, it's a smaller size population that is in decline. So to be vulnerable, you have to be less than 10,000 individuals. To be endangered, less than 2,500, and critically endangered, less than 250. So there are these big ranges within each. So we don't have to be, we have to be as accurate as we can be, but we don't have to worry about, oh, is it 9,000 or 9,500 or 8,500? We're confident that it's less than 10,000. Okay? And then, as I said, that with C, there's also something about that it's in decline. So is it declining 10% in over a particular period of time, or 20%, or 25%? So you need to meet both of these thresholds to be classified as vulnerable. So, for example, to be classified as vulnerable under Criterion C1, you'd have to have less than 10,000 individuals, and it has to be declining by 10% over a set period of time. For D, remember D is just very, very small population size. So is it less than 1,000 individuals, less than 250 individuals, less than 50 individuals? So that's how the criteria or the categories and the thresholds work. So it's pretty straightforward. Where it gets a little more challenging is interpreting the criteria, interpreting how one counts the number of individuals, how one determines what length of time does one assess it for. And so that's what we'll go to now. So again, remember the data that we need is information on the distribution. We need to estimate the population size. We need to have some idea on how that population may be changing over time. Over what time? Is it a long-lived species? Is it a short-lived species? Then again, what the threat is, and if possible, some ideas for solutions to those threats. And we're going to be using the recommendations that Anders and I came together in this paper. So remember I said that red listing is a way to estimate a change in population size over time and use that information to infer the risk of a species going extinct. So obviously, population size is a key component of this. So how does IUCN define population? And you'll see that population for IUCN red listing is somewhat different than the definition you'd use in your population biology class. So for IUCN red listing, a population refers to the total number of mature individuals. Okay, total number of mature individuals. And a mature individual is defined as individuals known, estimated, or inferred to be capable of reproduction. Okay, so species that are never going to reproduce are species that are so old they no longer are, not species, individuals that are so old that they're no longer going to reproduce are now counted as mature individuals. And you're saying, oh, how do we know this? Well, the population size is almost always estimated or inferred. And this is done with all taxonomic groups, even birds and mammals. So there might be a few species of primates where they've gone out and counted every species of this monkey. But in almost all cases, that there is a level of estimation for what's the total number of mature individuals. And with fungi, of course, is challenging, right? Because the biology of fungi. So, as we all know, fungi, for the most part, are clonal organisms. So you have this clone, this genet all the mycelium here is one genetically identical individual. But over time, this individual develops and separates and can get separated into multiple parts. So this genet separates out and forms multiple ramets. So each one of these ramets are genetically identical to this original genet, but each one of these ramets are capable of producing sporocarps. So as we're counting mature individuals, we're thinking of each of these ramets as a mature individual, okay? So even though they're all genetically identical, so they're a genetic individual, but functionally, they're acting as separate individuals, because if this goes away, this one can still produce sporocarps, or this one can produce sporocarps, right? So we're really counting functional individuals. So we're counting ramets. Now, mycologists have a tendency to say, oh, woe is us, we have it so difficult because we have clonal organisms. But in reality, many plants and some animals are clonal, right? So how many individuals of this moss species are here, right? This is mainly a clonal organism. In the west coast of the United States, the western US, we have this tree, Populus tremuloides, which forms great clones. So probably this whole hillside is one genetic individual. So both the moss people, the biologists, and the plant people have had to come up with ways to count mature individuals. So mycology isn't unique in this situation. Understanding how to count clones is a problem. If you're, from an animal standpoint, right, corals, big coral reefs are made up of a clone of and divided up ramets of an individual. So Anders and I just came up with some definitions, and these were based, there's some recommendations, and these were based on the data that was available back in 2010, 2011 on population biology. There's been some new data coming in, so we're going to be looking to see if we revise this in the next couple of years. But for now, these are our recommendation, and these recommendations are approved by IUCN. I need to emphasize, if you know better data for the species that you're working on, you use the best data available. So if you're assessing species X and you have data, population biology data on that species, use that information. But if you're assessing species Y and you do not have that information, you can use the recommendations that Anders and I came together. And so I won't go into detail here because you have it in your handout or the material that we put online. But basically, say for lichenized fungi, if it's growing on a tree, a log, etc., each patch is counted as 2 mature individuals. We figured that there's been some dividing up. And if it's a continuous patch, so it goes for a long period of time, every meter squared is counted as 1 mature individual. For mycorrhizal species, we're saying that, hey, a genet is probably limited to about 10 meters in area. But we know that that genet probably is divided up. So we're going to count a number of ramets. And that depends on the species. So a lot of boletes or some amanitas may only put up 1 or 2 sporocarps. So for that, we will assume fewer number of ramets versus a marasmius that may make a carpet of sporocarps over that 10 meters. And there we might say there might be up to 10 ramets within that in that area. So it becomes from experience, but also just looking at the distribution of the sporocarps across the landscape. So again, we would use this to try to estimate how many individuals are here, and I probably would count this as at least 5 individuals. So again, that's how we count individuals. So how do we then estimate the total number of mature individuals? Remember, it's defined as individuals known, estimated, or inferred. So it's not just the number of Fungarian records. It is the number of Fungarian records times the number of additional mature individuals that you as an expert on that group think occurs. So we actually estimate the total number. So what we'll do is we'll say, okay, how many sites do we think this species could occur? It's known from 4 locations now, 4 sites across here, but this habitat that occurs continues for another 1,000 kilometers. So if we take the data we know at how frequently there are sites in the areas we've studied, can we extrapolate, can we estimate how many additional sites there may be through its habitat? Okay, so that'll give us a number of estimated sites. And then at the sites that we know it, how many mature individuals on average do we see? We normally see 10 mature individuals at this site. And we've just said there's now 50 sites. So we would say there's an estimated 500 mature individuals. Does that make sense? So we're doing a 2-step. We're taking our data that we have from our Fungarian information, from our collecting information, from other people we've talked to, and said, okay, here is where we know the kind of habitat that this species occurs in. How much more habitat is there? So we can estimate, we can predict how many more sites there may be. And at each one of these sites, how common is it? We only ever find it like maybe 1, 2 mature individuals we estimate per site. Or at each site, it's really abundant. So there are 20 mature individuals at each site. Whatever that is, we will multiply that out to come up with an estimate of the total number of mature individuals. So hopefully this graph will help. So we've identified this by kind of detectability. How easy is it to see this species? Is it a bright orange bolete that's 10 centimeters tall? Or is it a little brown hypogeous fungus, right? One is really easy to detect, really easy to see. One is difficult. And so likelihood of the easy to see, the likelihood that we've missed it is low. For something that's difficult to detect, the likelihood that there's more out there that we haven't seen is high. And then what is the area of appropriate habitat? Is it only known from 1 or 2 beaches on Santa Catalina Island? Or is it known from the entire Atlantic coast forest, right? If it's a small area, then we have more confidence that we have sampled it. If it's a large area, we have much less confidence that we have sampled it. So if you have something that's very distinctive that we are confident that's a small area, you only have to multiply by a small number. But if you have something that's very indistinct and it occurs over a large area, you need to multiply by a large number to estimate the total number of mature individuals. And then you also take into account how much effort. Was it collected, once by the original field site and no one's really looked for it again? Or this is something that we've been really interested in. We've had teams of mycologists looking for it for the last several years. So that intensity of sampling needs to be included as you're estimating the total number of mature individuals. And from all the workshops we've been involved with, this is the single hardest thing for people to be comfortable with, right? What is that right number? How much should we multiply it? How much do we really know about that species? So this is probably the hardest decision that one makes in the review process, but it's super important. And one thing we found is almost always we're underestimating how many species are out there, that you need to multiply by even more than you think you do. So for example, in the United States, we just have a project that's going on in California where we've asked the amateur mycologists to go out and search, and we gave them 10 species to look for. 8 of the species in just 3 months found a number of new locations, a number of new individuals. So just in 4 months, we greatly increased the number of mature individuals that we know of and the type of habitat that occurs in. But for the other 2, they didn't find it at all in those 4 months. So it's still pretty sure that that's a very rare species. Okay. So we've come up with an idea of how many individuals we think occur. We've estimated the total number of individuals. I said the other thing is we have to say, how is that population changing over time? And we can do that one of 2 ways. Occasionally, we actually have data on the fungi themselves that we can make a statement. So it's something, a species that is known from such a small area and it's intensively being sampled that we can actually have counts and say that, hey, 10 years ago, we had this many individuals that we find, and now we have this many individuals. Or it is a species that's sold for food in the market. And you say 20 years ago, they were selling, a half a ton of this species in the market. And now, even though it is still being searched for, they're only finding many fewer species are on the market. So those are real data that we can use to estimate the change in the population size. But more often, we're making these inferences based on other data. So loss of habitat or loss of the substrata. So for example, in this case, this is a map of Sweden. And so between 1983 and 2003, you can see that this light blue habitat type has greatly declined by at least 50%, let's say. If you have a species that is restricted to that light blue habitat, you have pretty good data that that species has drastically declined as well, because the amount of area, the amount of habitat, the number of sites that it could occur in has declined by 50%. Okay, so we're using habitat information to infer the loss of habitat more or less can equate to the loss of population size. Same thing is if a species is restricted to a particular host tree or something like that. If that host tree is under great decline, then we can infer that the fungus is population is under decline as well. I'm going to skip this one. So that's how we count number of mature individuals, and we estimate and infer population decline. And so that's necessary for Criterion A and C. Criterion B, and D. Criterion B is the only one that uses area. So what is the distribution of the species? And so to document the amount of area a species occurs, there's two different ways to do that. One of it is called extent of occurrence. The other is area of occupancy. Okay, so the extent of occurrence is, here are all the dots of the estimated sites or estimated mature individuals of the species. So for this one, it's distributed like this. This one is distributed kind of here. There's an outlier up here. And to calculate the extent of occurrence, we just draw a polygon around all of the mature individuals and calculate the area of that polygon. And that gives you extent of occurrence. So it includes a lot of area where the species does not occur, or we don't think this species occurs. And you say, why does it do that? Because if you're thinking about it, the fact that this is isolated actually means that if this part of the population is being negatively impacted, there's something going on here to threaten this, this part of the population isn't being impacted by that threat. So we're looking at the total area that the species occurs in. Area of occupancy is actually talking about how much space does the species actually fill. So in this case, we take those known and inferred distribution and we overlay it with a 2 by 2 meter grid. And we count how many 2 by 2 meters, so 2 square meter or 4 square meter grids does that species occur in. And we just total that number, and that gives us the area of occupancy. Okay, so this is based on 2 by 2 meter grid. Extent of occurrence is over this whole area that's under the polygon that encloses the distribution of the species. Another criteria another definition that's being used is severely fragmented. So severely fragmented means that it's a relatively small population, and each of the subpopulations, each of the smaller groups occur in small subpopulations that are distant from each other, so there's limited gene flow between these. So this is severely fragmented. This one, even though these subpopulations are distant and there's probably not gene flow between them, it's not severely fragmented because most of the individuals occurs in this one subpopulation. Okay? And the last, I know I've been giving you a lot of data, but it will make more sense when you start doing the assessments. The last thing is generation length. And that is, we're talking about how much time do you assess the amount of change for a particular species. So it's really talking about the turnover rate of the breeding individual. Is it a long-lived species? Is it a short-lived species? And so IUCN gives this, Mike, that you do assessments or you review kind of changes over time over 3 generations, or if it's a very short-lived species, say an annual species, it's over 10 years, or if it's a really long-lived species for a maximum of 100 years. And so just some non-mycological examples are, if you think of this field mouse versus this elephant, they're responding to threats, say drought or a fire or something like that, very differently. Or this viola versus this redwood. Short-lived species, a couple of years of severe drought or a couple of years of continuous fire could wipe out this whole population, versus something like this big tree would persist because then when that drought ends, it can go on doing its thing. Or the elephant can withstand shorter term disturbances than the field mouse. So having a general idea of how long a species persists is important for estimating what is the impact of the particular threat. And again, we've made some recommendations, and a lot of it, if it's on something that's growing on wood or something like that, is how long is the substrata going to persist? If it's on a piece of, on a tree trunk that's very hard, and we know that that tree trunk persists for a long period of time, we're going to say that the generation is longer than if it's on a piece of leaf litter or a piece of animal dung. So we've made these recommendations for how to, what to use for the 3 generations. So I've gone through this quickly. And just to give you an idea of how we interpret these IUCN definitions and how we use those in doing red listing. We're going to take a break pretty soon. We'll come back and actually practice. But I want to leave you with this. If you're interested and you want to know more, I strongly recommend this course that's online for talking about how to do red listing. It's really a great course called Evaluating the Risk of Extinction of Species Utilizing the IUCN method, red list method. And it's in Spanish, English, and French. I don't believe it's in Portuguese yet. But it's a great tool. There are these, 6 different modules, 7 different modules. And then you take a test after each one to show that you've passed it. And then a final exam, and you get a certificate that says that you've passed this, and you're now a certified ability to do red listing. So I strongly recommend taking this class. And here is the link to it. Basically, it's just conservationtraining.org. Okay, so I'm going to stop sharing, take any questions you have now, and then turn it over to Ricardo and Ida for next steps. So thank you very much.
Thank you very much, Greg. Let me see if you have questions here. Greg, there is a question from Mona Lisa Ribeiro.
Mm-hmm.
How can you know if a species is in danger if we don't know the species?
Correct. Those would fall into the place there. So one of the reasons we don't have more species on the red list now is that a number of species, we just do not have enough information to be able to do the assessment. The taxonomy is so confusing right now, or we don't have enough information on the distribution of the species. And in those cases, we just don't do the evaluation because we don't have the information. So, so that's my response, is that we just can't evaluate everything right now because we don't have the data.
Basicamente, se não tem como delimitar a espécie, você não consegue avaliar o tamanho populacional dela, entre outros dados. Okay, Mona Lisa. Greg, there is another question from Mona Lisa.
Mm-hmm.
How I know if the individual fungi is not capable of a reproduction?
I didn't understand that. Can you repeat, please?
How I know if the individual fungi is not able to reproduction?
Oh. Yeah. So one of the, and this becomes part of the issue we now have also with environmental sampling. We're finding, using DNA data, that some species, we're finding mycelium or spores much further away than we've ever seen these species reproduce. So we need to interpret, is that really, are the conditions correct in those habitats that these will ever be mature individuals, right? So we are, again, we have to rely on the evaluators as the experts of that species to be able to interpret, is those, are those places where those are occurring, will they ever be able to reproduce or not? So, yeah, it's an interpretation.
Okay. Thank you.
I hope I answered that question.
Alguém poderia verificar no chat do YouTube se a Mona Lisa está satisfeita com a resposta?
Había una pregunta de líquenes, ¿no?
So we have like a question? Sure.
Yeah, it was a question about lichens.
Yeah.
Like how to calculate the minimum area or something.
Yeah, why did we use the 1 meter square for individuals? That was based in our conversation with some lichenologists who made that recommendation. I don't know that we have good population genetics data to support that, but that was the way that the lichenology community was using it. So we just followed their recommendation. Not a great answer, but that's just that's the reason, is because that's what was being used.
Yeah, I would like to say that we are just learning, no, how to work with the different kind of organisms depending of their biology. That's why there are just like suggestions, but on the way we will start to understand how to do this approach in a better way. You know, it's just, yeah, we are learning, no? Okay?
Mm-hmm. Yes, we're learning. And, you know, for many, we use the best data that's currently available. So I to say, if you think of it, when we were doing phylogenies of fungi. So 20 years ago, it's more than that now, 25 years ago, when we started doing DNA phylogenies, we were making these phylogenies, these estimates of evolutionary relationships, with part of the ITS region on very few samples. We still published those, even knowing that we had hoped that we would have more data in the future. And now we're doing phylogenies, you know, on comparative genomes. But we didn't wait 25 years to be able to start making those hypotheses on relationships among fungi until we thought we had these genomic data. And we see that sometimes we didn't get it quite right, but oftentimes we had the general idea. So these assessments are hypotheses. They are best understanding based on the data that we have available now, with the expectation that as we have more information in the future, that may change. And it's not unique to fungi. They are changing bird listings as more information comes about, or mammal listings as more information comes about. So it's not unique to fungi. Every group of organism that the evaluators are using the best data they have available at that time. And we should just think of these as hypotheses. The best hypothesis that we can make, but it's still a hypothesis, recognizing that it likely will change. I see just from Mary Lise that Diago and Larissa have questions, but I can't find their questions in the chat. So do you guys want to ask the question just live? It might be easier than me trying to find it here in the chat. Larissa, hi.
Hi, Greg. Hi, Greg. Hi. Nice to see you.
Good to see you.
Greg, I have a question about generation length. Mm-hmm. Because, I'm most interested in gasteroid fungi, so stinkhorns, phalaenus. So it's a problem because the fructifications can be there for 2 or 3 hours, maybe. And then when you say that, how am I going to estimate that? Because you said that soil fungi maybe 10 years or 20 years, so I'm taking into account the mycelium, right?
Yes.
But for example, for the phalaenus, what I can say is that I noticed that when the environment is disturbed, the mycelium is very short, long term. For example, here near the camp, our university, sometimes I can follow the fructifications, but then I know that they cut the grass, the lawn, and they have using chemicals, so the mycelium is dead now. So should we differentiate if the specimen is found in a disturbed or not disturbed environmental? I don't know if
right. Depends on that particular species. So if it's something that you find most often in disturbed habitats, that will be different. It also the trick with soil fungi is we don't necessarily know how specific what they're growing on. So is the food source, when the food source is all used up, that mycelium is going to stop, right? So the reason we gave the longer time limit for those is we assume that mycelium will keep growing, looking for, if there's nothing to stop it, it will just keep moving and looking for new food sources so that that rammet can persist for a longer period of time versus something that's growing on a piece of down wood. When it uses up that piece of down wood, it's done. It hasn't moved. There's not additional resources. And so that's why we look at these mycorrhizal species or soil inhabiting fungi as the ability to live longer because they don't have anything that stops them from growing and looking for new food.
Mm-hmm. So this data that you have that maybe 10 years or 20 years that the mycelium survived is based on literature or some estimation?
It's based on some literature on trying to estimate how long the litter survives. So it's not based necessarily on, we don't have good data on how long the mycelium is there, but we have data to say that, if it grows on this type of litter, in 10 years, that litter's gone. So therefore, we assume that species, that individual is gone.
Okay.
So if you think in a more extreme case, if it's a species that grows on animal dung, right? Once that animal dung's gone, it has to sporulate and go to another piece, right?
Yeah.
So that is very short-lived. And so
but then
we
you
figure
have the clones. Yeah.
Right. But that clone doesn't move from, it's not growing by mycelium from this piece of dung to this piece of dung. It grows, it sporulates, and the spores go to, so it's starting a new individual.
I see. Okay.
Versus if you have leaf litter, it goes from this leaf to this leaf to this leaf to this leaf, and it can keep growing.
Mm-hmm. Okay. Thanks, Greg.
Yeah, but that's again why, why we don't just take, we don't have a computer do this work, right? That just goes to, okay, here's the Fungarian records, calculate this. We need the experts who understand more about the biology of the species that they are working on to make those informed decisions. So based on your knowledge of that species. So these recommendations I'm giving is a general recommendation. But if you know something about this species and you think that really doesn't fit this species, I can make a better estimate, do so. And Thiago, you had a question.
So, hello, Greg. First, there is a question of Mateus Serafim. His question is it possible to extend the lifespan of a shorter generation fungi if it switches or access a new host, example, discrete substrata to soil or wood?
Yes. So when we were talking about those short-lived species, those were for things that would be on what we called discrete, so on very separate pieces of substrata that there's nothing that it can climb to. So again, it's like I was mentioning with Larissa, if there is, a species that forms rhizomorphs, for example, if it's growing on a log, but it can form rhizomorphs and go to another log, that would not be on a discrete substrate, right? Because it could continue moving from log to log versus something that it's on this log, and when it uses up all the resources of that log, it's done. So that's why we differentiate between discrete or separate substrata versus more continuous substrata where the fungus can continue to move and look for new food.
Okay, I have two questions, a little bit different. How can we transform the habitat or host loss to population reduction? Is there a recommended ratio?
Again, I think it partially depends on what you know about the species. We are now starting to pretty much say if there's a 50% loss in habitat, there is up to a 50% loss in the population size of the fungus. So we're pretty much using a 1-to-1 ratio between habitat loss and loss of the fungus.
Okay, thanks. This one is a little bit more specific. Could you please explain the biology behind the numbers of the criteria C2, A2? Or is this too specific? I just can't.
No, I can do that. I'm just making sure I have it in front of me here. C2, A2. So C2, remember, is we have a small population size that is declining, but it is not only declining, but either all of the species are in one population or it's severely fragmented. Okay, so it has to be both. Okay. And the biology of that is that even though it may be a small population, if those populations are scattered across, there is some protection, I guess you'd say, or some reduction in threat, because if this small population is lost, you have these other subpopulations that still persist, which is why the question is, if this is a small number and it's all in one subpopulation, if something happens to that subpopulation, it's all gone. So it is taking advantage of the fact of how are those individuals distributed across the landscape. If it's distributed more broadly, it's basically protected from single events, versus if it's clustered in one place, it's all wiped out if there's a bad event.
Yeah, it seems reasonable. But as far as I can see here, if you have 100% of the individuals in one only subpopulation, the species is vulnerable. But if you have 90 to 100%, the species is critically endangered. So it seems a little bit odd to me.
That's just because you have smaller number. Remember, it goes together. You're also looking at the total number of individuals.
Oh, yeah.
Right? So the smaller
right.
number of individuals, you've increased your chance of going extinct anyway.
Yeah, sure.
So we can reduce
you
the
need
threshold.
To think about the two things.
Right. So we can reduce the threshold of the number of individuals that are in a subpopulation.
Yeah, sure. Makes sense. Thanks, Greg.
Yeah.
Hello, Małgosia from Poland. Hello, Greg.
Hello, good to see you.
Thank you. I'm not visible, but anyway, I would like to ask you about your opinion. What do you think about the using of environmental DNA data in categorizing or looking for threat to fungi? Maybe you could tell us if there are some this kind of studies that we could use as a model or just something done this way, because I looked for it and I didn't find any example.
Yeah. So as I said, I think, yes, I think environmental data is very important, but we have to use it carefully, because again, we're looking at for red listing for mature individuals. And we know that, so environmental samples can greatly expand where we have information that a species might be occurring over some short period of time, but we don't know if that species has become established, right? Is that species or the conditions of that at that site going to be correct so that it can actually fulfill its whole life cycle and reproduce? If you think of the same way as, say, a seed bank, you can go out and find seeds of a plant, or even sometimes young plants growing far away from where the main population is. Birds carried it or whatever. But it's basically that species is experimenting, and it may not persist, it may not live. So from that standpoint, I use the environmental data to say, hey, this might be there. We need to study those further sites more. So it gives us good information on where we need to be looking. The other place that has been very successful, though, and this is Gareth, why am I blanking out his name? From Scotland, I'm blanking his name, has been using environmental samples for within areas that we know that it's the range and the habitat for the species, but we just never found it. And so has gone in and confirmed the presence of this species in places that it probably should be. And so then we can use that information and say, yeah, I believe that. So I think it's in both ways. So Gareth's data, I think, are very successful. The other data are just, I think we need to follow up and study those extended distributions more. Does that help? Does that make sense?
Yes, thank you very much.
Sure.
I just wonder what is your opinion, because it's it's still emerging, the role of the DNA sample. So it's important to think about it also. Thank you very much.
Yeah. So another example where environmental sampling is very interesting. So one of the species that's listed as, I think it's endangered or critically endangered, is this very large polypore fungus that's restricted to Western North America, Brigidoporus nobilissimus. It's one of the largest fungi. It can be several meters in diameter. It is perennial, occurs, and it only sporulates, it only forms these sporocarps on large old growth trees. Okay? But when we've done environmental sampling, we find the mycelium in many trees all around there, in small trees. Okay, so it's there growing as an endophyte or something like that, but it's not sporulating. But we know it needs these big trees.
Yeah.
And we know with our current forestry practices and climate change, the chance that any of those other trees are going to get to be the size to be a fruiting body, from an IUCN perspective, we don't count all those little trees. So the mycelium's there, but they're not mature individuals, because we can't predict, or we can predict that most of those trees that we find it in are never going to get to the size and the age that's going to allow that individual to sporulate. So that's another example of how we have to be careful how we use these environmental sampling.
Yes, so this is a very good example. And by the way, it is quite a simple example to consider, because, I'm interested in threat to small fungi, just like micromycetes, mats, and so on. So this is not so simple in case of them, because there is not such a threshold of the substrate this way. But anyway, thank you very much.
Sure. And I did see another question about that. Oh, I guess it's Tina that asked a question about these plant parasitic fungi, so microfungi. So we've been talking about that, and we don't have as good of information to use some of the criteria. I think the main criterion we can use for microfungi now would be Criterion B. Say it's a host-specific species, and the host has a very limited distribution. You would use the distribution of the host for the distribution of the fungus. So Criterion B would work. And Criterion A could work, is if you have it's a host-specific fungus, and that host is declining very rapidly. The thing to be careful about in both of those is the generation time, right? So if the host is a long-lived tree, so the decline of that host might be over a long period of time. But your microfungus, your plant pathogen, is an annual species. It's a rust or a smut that turns over every year, or a small ascomycete. You'd be assessing the change over 10 years. And so do you have a sufficient decline in your host over 10 years to be able to list it, or to be able to say it's vulnerable or something like that? So that's how you would use these small microfungi on plant pathogens. What else? See, there's a whole bunch of questions. Yes.
Hi, it's me. Maria.
There you are. Hello. Okay, you're too loud for your face to come up. I said, I know that voice. Who is that? Yes.
Emma here. So I wrote a question because I don't know if you have an input about like how herbicides and poison that are applied, for example, in golf courses affect the fungal soil. And I ask that because in Brazil, we have a lot of areas where the pesticides are sprayed by airplanes, and we obviously don't have anything fungal related in our legislation, and I wonder how that's going to be affect the fungi here in these areas, because like I've been like to limited times to Florida in golf courses, but I've never seen a mushroom in a golf course.
That would fall into the whole criteria of more of the data I know have been looking at other kind of pollutants, so nitrogen deposition, things like that. I would assume that fertilizer we know can negatively impact mycorrhizal fungi, right? If you have a lot of fertilizer, something that's going to reduce the amount of infection of the mycorrhizal fungi. And a lot of these chemicals are fungicides. I mean, on the golf courses, they're actually putting on fungicides. So we know that is impacting that. The question would be, from a conservation standpoint, are these golf courses the major habitat for those species or not? Is there drift? Is the wind blowing that those fungicides, those insecticides into the natural areas enough that it might be impacting the natural populations of these species? So again, it's going to depend. Or being carried to water resources. So it very much could be. I don't know of any.
Like nobody's looking at that, probably.
I don't know anybody's looking at that from a conservation standpoint. You know, they've looked at that from, as I said, nitrogen, other pollutants they have looked at, right? There was a lot of work that was done on acidification, so nitrous oxides, things like that, acidification. There was, in fact, that's what started much of the fungal conservation work was in Europe, where they saw these declines in sporulation due to either acid rain or nitrogen. So the first, this was back in the 1980s. So some of the earliest work on fungal conservation was because of pollution. But I don't know anybody who's looked specifically at pesticides.
Right. But that would be
I'm sure it has an impact. All
right. Thank you. Thanks for the talk also.
Greg, there is a question by Lara First. She wants to know, she's asking you to talk a little bit more about how to translate an AOO or EOO into a number of sites. Always find that really difficult to do.
So it's the other way around. You're not translating EO or AO into the number of sites. You're taking the estimated number of sites and basically plotting those dots. And then for the EOO, drawing that polygon around that, okay? And that gives you the extent of occurrence, okay? So the first step is to have those dots that says this is the distribution of the fungus. And so with that, you may not have all the inside dots. You just have the extreme dots is all you need for that. It's here in, this spot and this spot and this spot, and you can draw your polygon around that. That's all you really need to do EOO. For AOO, much more difficult, right? Because then you would need to actually have your dots for each of your estimated mature individuals, and then lay over that 2 by 2 meter grid over that and count how many of those grid cells it occurs in. Okay? So that's the way that works. So you need a lot more detailed information to do an AOO than an EOO. For the EOO, you just need to know you need to know the extremes of the extreme edge of your estimated population distribution. Did that answer that question?
I believe so.
Yeah.
Thanks.
Okay.
I mean, I don't know. I can't answer for Lara, but I thought it was very clear. Thanks.
Greg, I have maybe a comment about that with the polygons. I think that it's very important not only to do the polygons, but also to consider the habitat. Because sometimes, for example, here in the Cordillera, the forests are like in a line. So you cannot just cross and do a polygon without understand the geography, the habitats where are located. So I think that this is very important just to consider when the people is using this approach.
Well, for EOO, for the extent of occurrence, one doesn't look at habitat or where it could or could not occur. The EOO is just over. So if there was a species that occurs in Florianopolis and then a little bit along further up the Atlantic coast, and then in Lima and in Bogota, your polygon would cover that entire area. Even the fact that there's all kind of habitat in there that the fungus is not going to occur in, okay? Because remember, EOO is just measuring that entire area. And the reason for that is because your mature individuals in Bogota are going to be under different threats and probably under a different intensity of threat than those species in Santa Catarina Island. And so we're discounting all that habitat in between. We're really just looking at how broad that species is distributed. Now, when one is then calculating threat or calculating population decline, one needs to think about the habitat, right? So we've had this in several cases where we know it's declining high, say in the bulk of the most of the species probably occurs, say, in Brazil, in the coastal forest. But there is a few samples, and we think of a small part of the population is in the Amazon. Well, we would then look at the percent decline in both of these places. And if most of the individuals are in a site that has a higher decline, we would use that higher estimate of decline, right? So again, that's why we have experts doing these assessments. You need to take the data that you have and make these decisions. Oh, that's right. If you didn't see that, Drew Simmons shows that we do know that these pesticides and fertilizer has been important in these semi-natural grasslands in Europe, which are sites for a lot of the wax caps, a lot of the Higroscites. So that's one area where it has been studied a lot.
I have a question.
Mm-hmm. Sure.
I have the same question Leo asked a few minutes ago. I really don't understand under Criterion C. I would assume that if 100% of the individuals, or almost 100%, are in just one subpopulation, I would say the species is more endangered and not less endangered. So it's like the other way around. Am I wrong?
It is more endangered, but you have a larger, so if you look at the change in across, if you look at this, so you're looking at C2 again. So if you're looking at how many individuals are in one population, that declines.
Okay.
As you have a smaller number of individuals, right? So the fact that for critically endangered, you only have 250 individuals, you've already increased your chance of going extinct, so you can reduce your stringency, your threshold for the number of individuals in a subpopulation, right?
Okay.
So they're going hand in hand. When you have 1,000 individuals, or 10,000 individuals for vulnerable, then you need to be more restrictive for the number of individuals in a subpopulation, because you just have a bigger population, so you've reduced your threat.
Okay, so we must understand that in a column. So if this is the number of people, perfect. That makes sense.
Right. Yeah.
Thank you very much.
Right. So you could have 250,000, you could have 2,500 species that's endangered, but less than 95% of them being in one subpopulation, and you couldn't call it as endangered, right? Because it doesn't meet, it has to meet both thresholds.
Perfect. Thank you.
Greg.
Yes.
There is a question from Leonor Costamaya. Do you know Leonor?
Oh, great. Leonor. Yes.
She wants to know, what about soil fungi, like the arbuscular mycorrhiza fungi?
Yes. I have to say we have almost, I won't even say almost, I don't think we have any data on those group of organisms, unfortunately. Right? There is still such a confusion, and now Lenore knows more than me, that there's still confusion on what is an individual, how those species are distributed across the globe, right? If there really are there 350 species globally? I don't know. So we haven't tried to do anything with arbuscular mycorrhizal fungi. I apologize, but I don't know how to do that. So I could just say we don't know. It'd be great for somebody to look at that. It's a good PhD project for somebody to do that. Sorry.
There is a question that Zhu did in the name of Dirce. Where is it? So Dirce is asking, what would be the anecdotal data to estimate the number of localities and functional individuals?
For Did she say what group of organism? What group of fungi?
Well, she works with macrofungi and actomycorrhizae.
Yeah.
She didn't say that, but I know who she is, so I assume.
Yeah. I think we start with what has been documented. What does the literature say? What's in the Fungarium data? Because that gives you an idea of what the potential habitat is, what we know about the distribution, and you can also infer from that as how much effort has been used to try to find it, right? So if so we start with that. And then again, we'll say that this is a species that occurs in a very restricted type of habitat. So for example, which is part of a paper that Maria Elise led for this really very distinctive species that is only known from Santa Catarina Island. So all of a sudden it's a distinctive mushroom from a very specific habitat. So it's possible to, estimate that we have a pretty good idea of the number of mature individuals and the distribution of that species. Versus something that is much less easy to collect or to identify. It's it's a white resupinate fungus that grows on a variety of different hosts across the Atlantic forest. We would have to infer that there are a lot more individuals there than we have identified, that we have collected. So again, it depends on what we know about the species, how big of a habitat it may occur in, and how easy it is to see. So how likely it is that we've collected it, or how likely it is that people have walked over it and didn't collect it. So it's that combination of factors that go into that. No easy answer, except for the fact that in almost no case can we say we have identified, we know every single individual that's out there. We always have to multiply some. And if Anders Dahlberg was on the line, so from a place like Sweden, which has been sampled intensively for mushrooms for 300 years, they are still finding new subpopulations of species of these things after hundreds of years of sampling. So we always have to multiply, even in places we think we know very well. Okay. What else?
I see another question. I don't know if there's anything. I can just see what's in the chat, and that is one from Fungal Conservation. Hi, Greg. We are wondering about the sampling efforts and their time extension. How do you set the dots over time? Desculpa, Ricardo. Não sei se tu tinha algum.
Yeah, no, that's a great question. Partially it depends on habitat, right? So we will look at historical data, but if we know that many of those early records, those sites no longer occur, I wouldn't use those, right? Because those locations are gone. They were now part of Rio de Janeiro, right? They used to be on the outskirts of Rio, but now the city has expanded and we know that those sites don't exist anymore. They're not very useful. except for the idea of what that habitat was. So I can still use that information to predict where the current habitat would be. But I will only go back and use historical data as far back as those sites actually still exist. So I guess that's my answer, you know. So again, if we go to Europe, there's species records from Fries, from Elias Magnus Fries, who collected in this southern part of Sweden. But we know that the habitat type has totally changed over the 250 years since Fries collected there. So to say that something was collected at this particular site doesn't really mean that you're going to find it at that site now, because the habitat has changed from, I don't know if it was more pine trees and now it's hardwoods, or vice versa, but I know the habitat has changed a lot. So again, we need to understand what the habitat is, and are those sites still existent?
Rio is a good example, Greg, because for example, some of Corner's species that he collected here were in areas that are like highly thirsty, or that became slums over time, and they don't exist anymore. They're like houses and, you know, housing and trails and people everywhere.
Yeah. So you can't really count those records now because they don't exist. It's the same thing when I've gone back and tried to retrace Rolf Singer in your area, Francisco. He would talk about, you know, just going outside one of these cities and, you know, collecting. And we now have to drive very far because that whole area has been transformed by urbanization. So, yeah.
Greg, I think we do not have more questions for you, but I'd like to say thanks. Thanks for your time. Thanks for sharing your knowledge with us. It was so nice. Thank you, Greg.
Oh, my pleasure. And I will be around. So when I know we're going to take the break and work on the species and the subgroups, I'll be back or available to be part of the conversation when we reconvene and discuss the results of your subgroup work. So I will see you all later. So thank you very much for listening.
Thank you, Greg. We would like to take a photo of everybody. So if you don't mind to turn on your cameras, and we are going to do a print screen. Is that okay for you? Pessoal, se todo mundo puder abrir a câmera aí para a gente tirar um print screen para colocar no nosso Instagram, eu agradeço. Lucas, se puder colocar todo mundo aí na tela.
Olha, Juliano.
Right, bring your cat or whatever you have with you. Cat or children, I see that. That's great.
Dá para colocar todo mundo, Lucas?
Enquanto o Lucas vai ajeitando a parte técnica para uma foto, eu só gostaria de comentar que o nosso agora, 12h30, e voltaremos às 13h30. Mas, por favor, entrem nos seus subgrupos. E aí eu queria falar depois com os professores. Aira, Francisco, Melissa, me gostaria de falar com vocês um pouquinho depois.
E depois, para a discussão, a gente volta nesse link aqui?
Depois, para a discussão, voltamos para esse link do Zoom.
Ok.
¿Quieres hablar ahora con nosotros cuando en el break?
Sí, después de la sí, durante el break.
Just a
Y
minute.
lo siguiente por Google Meet. ¿Foi?
Not yet, I think.
Ah.
What a great, what a good-looking group of people. This is terrific.
Duda, você tá aí? Ah, that's on YouTube. Okay.
Já foi?
I'll take a print screen. Print screen. 1 minute more.
We can only smile for so long, Thiago.
Okay, that's okay now. Thank you, guys.
Great, thank you. So, question for Ricardo. Are we coming back to the Zoom for the full discussion after the go-to?
Yes,
okay.
Greg.
Okay.
Yes.